All right, hello everyone, it's Chow here, and today we're going to continue our discussion about signal transduction pathways, so let's get started. In terms of our signal transduction, there's basically two ways we can break it down. You have direct and indirect. In terms of direct signal transduction, that occurs at the plasma membrane, and the function is basically the receptor itself, so there's no intermediate molecules. So an example of this is you have a binding of some sort of a signal molecule to a receptor, and then that effector is going to be right within the membrane. In this case, an ion channel that's going to be opened. And you have influx of ions that's going to trigger a cellular response. By contrast, you can also have what's known as indirect signal transduction. And indirect signal transduction involves some sort of a secondary messenger that mediates the interaction between the receptor and the cell's response. So in terms of secondary messengers, we can have things like cyclic AMP, diacylglycerol, IP3, calcium, nitric oxide, etc. And in this case, we have the binding of a signal molecule to the receptor, and that receptor is going to be mediated by some sort of effector proteins as well as secondary messengers that ultimately will trigger the cellular response further down the line. So it's basically, are you going to be activating some sort of result directly within the cell membrane itself, or are you going to require some kind of messenger, intermediate, you can say, that ultimately results in some kind of a response later on down the line through mediation and transduction of various intermediate proteins? And that's kind of the big difference between direct and indirect. So in terms of secondary messengers, we can talk a little bit about a couple of them, but one of the big ones is cyclic AMP. So cyclic AMP is kind of made in this particular process. If we think back to that G protein coupled cascade, right? You have the binding of that signal molecule to a G protein linked receptor that's going to cause the activation of this G protein by converting, switching GDP to GTP. And the activation of that subunit is going to trigger this particular enzyme over here to start making cyclic AMP. And we'll find out that this enzyme is called adenylyl cyclase. So adenylyl cyclase is activated, it converts ATP into cyclic AMP, and the cyclic AMP is then going to go on and start causing effects further down the line, further down in the cascade. So the cyclic AMP basically has two major target types. It binds to ion channels in many kinds of sensory cells, and it also opens the channels. It can also bind to protein kinases that are within the cytoplasm, and that exposes active sites and starts protein kinase cascades. So it activates protein kinases. Now, if you think back to your basic biology, right, the fight or flight response is going to be upregulating your sympathetic nervous system and downregulating your parasympathetic nervous system. So in this case, some sort of a scary or triggering event occurs that ultimately leads to the release of epinephrine or adrenaline, and epinephrine is going to bind to an epinephrine receptor on the plasma membrane. That is going to cause a conformational change in the epinephrine receptor, which is then going to activate your G protein by adding a GTP onto it instead of a GDP. And the activated GTP heterotrimer subunit, usually like the alpha subunit in this case, is going to activate an adenylyl cyclase. The activation of the adenylyl cyclase is going to convert ATP into cyclic AMP. The cyclic AMP is then going to act as a secondary messenger, go further on in the cascade, and start activating enzymes like protein kinase A. After protein kinase A is activated, it can then further start activating certain enzymes like phosphorylase kinase. Activation of phosphorylase kinase is then going to cause the activation of glycogen phosphorylase. And the activation of glycogen phosphorylase is then going to convert glycogen into glucose 1-phosphate, which ultimately becomes glucose, and that can be released from the cell out into the bloodstream and be sent to where it needs to be, because in this case, you want glucose out and about. And it's very important to keep in mind that protein kinase A also causes the inactivation of glycogen synthase. Remember, you're in a situation where you want glucose, So this particular binding of one epinephrine molecule causes a cascade of events, but it's also important to keep in mind that the binding of a single epinephrine molecule can dramatically cause increased activation of the cascade. So every single step in the cascade will be dramatically amplified. And that basically sums up the cyclic AMP cascade as well as the epinephrine pathway. Hopefully that's a little bit of a review for you, but if it isn't, something good to study up on. Now, one thing that's also important to keep in mind is that in terms of the fight or flight response and the activation of your sympathetic nervous system, eventually you're going to need to downregulate it, right? Because you don't want to be constantly active and constantly having epinephrine flowing around. So you have certain enzymes to do that as well. One of them is phosphodiesterase, which converts your cyclic AMP into normal AMP. And the conversion of cyclic AMP into normal AMP effectively shuts down this pathway and downregulates the sympathetic nervous system. And then, of course, conversely, it starts upregulating your parasympathetic nervous system, so you can go back into this rest and digest phase rather than this fight or flight response. One note about regulation is that there are different ways that this particular cascade can be regulated. And we can see that there are protein kinases which become active or inactive depending on phosphorylation or dephosphorylation of the protein. There's a little bit of a caveat with this, but in terms of the epinephrine pathway, usually protein kinases who are phosphorylating will turn on the cascade, whereas protein phosphatases, which dephosphorylate, tend to turn off the pathway. Now, if you think to other types of pathways, that might not be the same. So for example, if you go to like your gluconeogenesis pathways or glycolysis pathways, depending on whether you're in a fasting state or whether you're in a fed state, that can also affect whether enzymes are turned off or on. And the turning off or on may also be either phosphorylation or dephosphorylation. So that's something very complicated and kind of tricky to keep track of. But in terms of the epinephrine pathway, this usually tends to be true. Similarly, in terms of G proteins, G proteins can be turned on or off depending on kind of the binding of either GTP or GDP. Usually the binding of GDP ends with the protein being shut off. The binding of GTP to the G protein will cause the protein to be turned on. Again, there's probably some exception to the rule, but generally speaking, GTP binding to G proteins causes activation of the G proteins. And then finally, we have regulation via cyclic AMP. So if you have adenylyl cyclase that's turned on, you can convert ATP into cyclic AMP. By contrast, phosphodiesterase can turn off that signal by basically converting your cyclic AMP into normal AMP, and that shuts off the pathway. So that's just something to keep in mind. Regulation can occur in many ways, but these three are kind of the big key factors in helping moderate your fight or flight response. It is worth mentioning, though, that there are other secondary messengers like PIP2. So PIP2 is this lipid-based molecule that is substrate for two secondary messengers, IP3 and DAG. And you have a certain enzyme called phospholipase C, which hydrolyzes PIP2 into DAG and IP3. And these products will actually cause many cellular responses, including like calcium influxes, as well as like NF-kappa beta translocation into the nucleus, and that induces like transcription of certain pro-inflammatory genes. But what's really kind of interesting is that overactive pathways can lead to excessive brain activity, which can be found in certain illnesses like bipolar disorder. So in terms of treatment, you can use things such as lithium salt, which blocks your G proteins and inhibits them, and it also can block IP3 synthesis. And so in doing so, that can actually shut off this pathway and potentially help treat over excessive activity of these pathways. Something to keep in mind. Okay, and moving on, we have other types of secondary messengers such as nitric oxide. This is an interesting one because nitric oxide is a secondary messenger that's produced by other secondary messengers controlling blood flow in the system. And NO2 is something that can be used as a treatment for certain types of illnesses like angina as well as ischemia. So if you are an ischemic patient and you have decreased blood flow to certain parts of your body, like your chest, that can cause hypoxia as well as chest pain. Now, you can treat this with nitroglycerin, which releases NO, and this NO, or nitric oxide, will go through this signal transduction pathway that ultimately leads to smooth muscle relaxation and vasodilation. And when you dilate your vessels, you allow more blood flow, and that can increase the amount of blood flow and ultimately increase the amount of oxygen within that region, and that can decrease chest pains. The signals then are going to cause some sort of an effect. There are different types of effects that can occur. It can cause changes in enzyme activity. It can cause the opening of ion channels, as in the case of olfaction and fertilization. And it can also trigger differential gene transcription. So we're going to go through these three, and hopefully you'll get an idea of what's going on. We're not going to go talk about too much in terms of enzyme activity, because that's exactly what the cyclic AMP epinephrine adrenaline pathway is. You have regulation of different enzymes via secondary messengers, as well as shifts and changes in the presence or absence of certain secondary messengers. And that can ultimately lead to increase or decrease regulation and activity. Alternatively, you can have ion channel changes, and ion channels can function as receptors and other components of signal transduction pathways, and they can also have an opening of ion channels in response to certain signals. Olfaction is a big one here, and then also it can have G protein responses that can open ion channels, as well as G proteins that activate adenylyl cyclase. Remember that big enzyme adenylyl cyclase, which converts ATP to cyclic AMP, and that cyclic AMP can then, as a secondary messenger, open ion channels, which lead to influxes of certain ions like sodium and calcium that can lead to stimulation of nerves to send signals to the brain. So that actually is sort of what smelling is, or olfaction is. You have a situation where you have an odorant molecule that's binding to an odorant receptor that's going to cause the activation, again this time, of a heterotrimeric G protein that's going to ultimately cause the binding of GTP to that particular G protein and activation of that G protein, and that one of the subunits is going to activate adenylyl cyclase. The activation of adenylyl cyclase is going to convert ATP into cyclic AMP, which then binds to an ion channel, which causes the opening of the ion channel, and the opening of ion channels will allow for influxes of calcium and sodium, and that is going to send a signal to the brain. And depending on sort of what odorant molecule is bound and the activation pathway, that's going to initiate a signal that's specific to specific areas of the brain, which then you, as the individual, can perceive that signal as a specific scent. Ion channels also play a pretty big role in fertilization. So once you have the entry of the sperm into the egg cells, you have a rapid increase in calcium concentrations. So one of the big problems is you don't want polyspermy. You only want one specific sperm fertilizing one specific egg. So in order to prevent polyspermy, your body has kind of a fast block that occurs with electrical changes, but also a slow block that occurs using calcium influx. So the influx of calcium concentrations can act as a slow block to prevent additional sperm from entering and fertilizing the egg, and therefore causing problems such as polyspermy. Now, in terms of signal transduction pathways, it can also lead to major changes like shifts in gene expression. So differential gene expression in this case can be what's needed for cellular growth and cellular division. So signals from growth factors binding to growth factor receptors can cause activation of various cascades of enzymes and proteins that ultimately lead to changes in cellular responses. So in this case, you have a growth factor that's binding to some sort of a growth factor receptor that's ultimately going to dimerize, and oftentimes these growth factor receptors have innate tyrosine kinase activity, so they have autophosphorylation, and that allows for the activation, the docking/activation of other types of proteins like your RAS signaling pathways. RAS proteins can then ultimately activate things like RAF proteins, as well as MAPK proteins that enter into the nucleus and then stimulate transcription of genes for cellular growth and cellular division. The big problem, of course, is that this is a very sensitive and balanced pathway, so if you perturb the system, that can actually cause problems like neoplasms and malignancies. So some cancer cells have an abnormal form of a G protein called RAS. So RAS G proteins are kind of like your alpha subunit of your heterotrimeric G proteins. And RAS, what normally can happen is RAS can kick off the GDP and bind a GTP. The binding of the GTP activates the RAS, like what occurs in your heterotrimeric G protein subunits, and that causes a brief stimulation of cell growth and division. Of course, the RAS can also hydrolyze that GTP back into a GDP form, and that causes it to become inactivated. You can also have other additional enzymes that can mediate this process and speed it up. But in certain types of cancers, you have an abnormal RAS that is constantly going to be bound to a GTP, and it's unable to kick off that GTP, or it's unable to hydrolyze that GTP back into a GDP. So that RAS is constantly active, and it's going to constantly signal those cells to divide and grow and divide and grow. And in cancer cells, that is one of the mutations and mechanisms that can result in severe malignancies. But what's really kind of fascinating about that is that because you have specific mutations within specific parts of the signal transduction pathway, you can actually develop drugs and therapies to target specific mutations. So rather than doing broad spectrum radiation or chemotherapy, if you are able to detect specific mutations within cancer patients, you can utilize drugs to target those specific patients. So while targeted therapies are not going to necessarily have as strong of a curative effect, they are going to have a significantly decreased amount of adverse effects. And because you're targeted specific mutations, you can be more selective in your toxicity against cancer cells while avoiding natural healthy cells. One of those include growth factor receptor inhibitors. So if you have a patient that has a mutation within their epidermal growth factor receptor, for example, you can actually use a specific drug that targets that epidermal growth factor receptor, downregulating it and ultimately shutting off that pathway and preventing increased cellular growth and division. So examples of that include like gelfitinib as well as osimertinib. Osimertinib is one of those drugs that's used for adenocarcinomas of the lungs, especially in non-smokers. And you can also have other types of inhibitors, including tyrosine kinase inhibitors. So examples of that include your JAK inhibitors, your JAK2 inhibitors, for example, such as ruxolitinib. Ruxolitinib is a particular tyrosine kinase inhibitor that's used for individuals with cancers such as polycythemia vera or primary myelofibrosis. You can also have T-cell surface receptor inhibitors such as ipilimumab, which is a CTLA-4 receptor inhibitor. And by inhibiting CTLA-4, you decrease the downregulation of T cells, so you stop them from being downregulated, you keep them more active, and therefore they're more likely then to start targeting and destroying affected cells such as tumor cells, while potentially avoiding healthy cells. And of course, there are many others out there as well. So this is certainly an area of research that has a lot of promise because you're able to avoid doing broad spectrum chemotherapy that might be affecting both the healthy cells as well as the cancer cells, but rather by being able to find specific mutations and target those specific mutations, you're able to decrease the adverse effects while increasing the specificity of the toxicity against those tumor cells. So I hope you found this at least mildly useful. It's a pretty complicated topic. There's a lot of stuff to remember, and different pathways, different cascades, I think are always a little tricky for students, but I would suggest going through your notes and try to kind of visualize some of those pathways and what's going on, and hopefully you get a little bit of an idea as to what to expect kind of going forward with this class, as well as maybe in the future when you're taking a biochemistry course or if you're deciding to go into medical school. This is kind of what your molecular and cellular medicine block is kind of like. So again, a lot of stuff going on. It's very complicated. However, there's a lot of good work that's being done in these areas involving signaling cascades, and perhaps that might be something you want to do in the future as well. So best of luck studying. Thanks for watching, and I will see you in the next video. Take care.